This is Alvin the Chipmunk. I'm here to talk to you today about the scientific method. How does one think like a scientist? I like to think of it as how do I solve a problem or achieve a goal? The first half of the scientific method begins with a question or problem. This is followed by the hypothesis. Next, the scientist designs an investigation. These words together give us the acronym CUFID. The second portion of the scientific method involves measuring, to record, to analyze, and finally to make your conclusions. These give us the acronym MRAC. Let's begin with the first part of CUFID, the question or the problem. Many times we run into questions or problems in our lives. For example, you may want to know which color of light will grow a plant the best. Oftentimes you can get help to figure out an answer to a question or problem by doing background research. And eventually, based on what you know or find out, you can propose a solution to the problem or an answer to the question. This proposal leads basically to a statement about what you think is the solution to the problem or the answer to the question. The hypothesis is really a proposal for a test or an experiment where you say that I'm going to set up this experiment, and if I do this one thing, it will cause this to happen. And it will happen because of something I think or that I've learned recently in my background research. Now this leads naturally to a structure for the hypothesis of if, then, because. For example, if I grow plants under 4 colors of light, green, red, blue, and yellow, then green will grow the plants the best because plants are green. Now that we've laid our cards on the table, so to speak, and we've made a hypothesis, now it's time to see whether or not it will be supported by the actual occurrences of an experiment. And we have to make darn sure that the experiment is fair and valid. You do this by making sure that your investigative design is correct. Let's take a look at the design of an experiment in more detail. Let's zoom in on trial number 1, experiment number 1, and talk about the independent variables. Independent variables are what we change in the experiment. Earlier we mentioned the color of light. We could have one sample batch, number 1 with red, number 2 with yellow, number 3 with blue, and number 4 with green light. And it's important to remember to have plenty of samples of each different type of independent variable. Controlled variables, on the other hand, are the things that we do to each sample batch to keep them exactly the same. That way we know that the independent variable is the reason that something happens. In our experiment with light and plants, we would have to make sure that all the plants, each sample batch, had the same amount of water, the same kind of water, the same soil, the same amount of soil, and so on. That way we will know that the independent variable is the reason something changes. And that change we're looking for can be measured in the dependent variable. In our experiment with the color of light and plant growth, we're looking for how much growth the plants will have based on the color of the light. As long as our experiment is well designed and that our variables are controlled that need to be controlled, such as the soil and the water and the amount of sunlight, then we can be sure that our results will be valid, that the reason our dependent variable changes is because of the independent variable. But sometimes some things can go wrong in experiments. For example, one batch might get some bad temperature or something might happen to it. So it's always a good idea to have more trials, to do the experiment more than once. In that case, if you've done it several times and your results can be replicated, done again exactly the same, then you know that your results are valid. But we're getting a little ahead of ourselves. Let's talk a little bit about measurement. There should always be some sort of measurement when you determine your dependent variables' changes. These could be physical characteristics such as electrical conductivity. The size could change, weight, or perhaps temperature. Mass and volume could change, which is density. And then when there's time, that can be measured as well as a host of other things. These measurements can then be recorded. It's important to record your measurements in a couple of formats. A table and graphs are always good to use. When you do put them into this type of format, then you can continue with your experiment to the final concluding steps. With good data on graphs and tables, you're able to make a good analysis of the data, which leads you finally to your conclusion and an answer to your original problem or question. And that concludes the scientific method. I would like to add one thing. It is important that you keep good records and take accurate measurements so that other people will be able to replicate what you have done. This is Alvin the Chickmunk saying thank you.